Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. 1. To help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2. To build a supportive community where we can exchange the most valuable experience-based research advice. 3. To share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Welcome to the deep dive. Today, we're plunging into really a groundbreaking paper from Nature.
It is.
It's completely resetting how we think about gene regulation. For decades, the central dogma, DNA is mostly passive, right? A blueprint.
Pretty much, yeah.
And transcription factors, TFs, they just bind individually. But what if? What if DNA is actually playing matchmaker, actively bringing pairs of TFs together, like a dance?
That's exactly it, and it's truly counterintuitive, isn't it? This paper shows DNA isn't just static.
Not just a template.
No. Its structure, its unique features, can actively recruit and assemble specific pairs of TFs.
Yeah.
It fundamentally changes the whole gene regulatory code. It's like a whole new paradigm.
Wow. Okay, so you've shared some incredible sources for this deep dive, and our mission, as always, is to cut through the jargon.
Get to the good stuff.
Yeah, get to the core insights, the surprising bits, those aha moments. You can really grasp why this work is so significant, so innovative.
Okay, so background first. For a long time, the view was, well, pretty simple.
Right.
DNA had the sequence. TFs, the transcription factors, they found their individual spots, turned genes on, turned them off.
Like keys and locks.
Yeah.
One key, one lock, simple.
Huh.
But things started getting fuzzier.
Precisely. Those high-throughput promoter screens, they started hinting at something more complex.
Combinatorial rules.
Yeah, rules that just couldn't be explained by simple one TF, one site binding. There was this nagging gap.
Okay.
We didn't know if DNA's own structure could actively recruit pairs of TFs, like pre-assembling the machinery, before they even get to the chromatin, or before co-activators show up.
And that's where it gets really interesting, because figuring this out has huge implications. You're talking decoding disease enhancers from GWS studies.
Exactly.
Maybe engineering better gene circuits for therapies. It's fundamental how cells get their identity, how they work.
It really is getting to the root of it.
So the researchers, they noticed something was off. Predictions of where TFs should bind.
Didn't always match reality.
Right. Didn't match where they actually bound. So the bold hypothesis. What if specific DNA shapes, like base pair geometries, act like docking pads?
Docking pads for two TFs at once.
Wow.
A truly innovative idea, but incredibly hard to test back then.
How did they manage?
It only really became feasible with advances in cryo-electron microscopy. Cryo-EM.
Ah, the resolution revolution.
Exactly. Lets you visualize these big multi-protein DNA complexes, almost down to the atom.
Yeah.
They focused on two common TF families, ETS and GATA.
Why them specifically?
Well, they're known to cooperate in blood cell development, hematopoiesis, and also in cancer. So, good candidates.
Okay, so they have the hypothesis, the tool.
Yeah.
How did they start mapping this dance?
They used a method called CAP-SELECT. It stands for Consecutive Affinity Purification, Systematic Evolution of Ligands by Exponential Enrichment. Bit of a mouthful.
Ha! Yeah, sounds intense.
Think of it like super efficient fishing for DNA sequences that specific pairs of TFs like to bind to.
Right.
And they scaled it up, adapted it to a 384-well microplate format, screened over 58,000 different TF-TF pairs.
58,000, that's massive. What did that huge screen tell them?
Okay, first big finding. Over 1,500 TF pairs showed preferences for specific spacing or orientation, or both, when binding DNA.
Spacing and orientation.
And mostly short-range interactions. Less than 10 base pairs apart for their characteristic binding motifs.
So really close together?
Very close. You see this with known pairs like HOXB13-1 and TED4 clock. Suggests a very precise arrangement.
So it's not just that they bind together, it's how close and which way they face. That matters. It sounds like DNA is dictating a kind of grammar.
That's a great way to put it, a grammar.
Mm-hmm.
More complex than we thought.
Definitely.
And secondly, they found over 1,100 new things. Novel composite motifs.
Composite motifs, meaning?
Meaning these weren't just two individual motifs sitting side by side. The sequence itself looked different from what either TF would bind alone.
Right.
Yeah. Take the FOXA1 ELF2 composite motif.
Yeah.
Truly distinct. And crucially, they checked this in vivo, in living cells.
Using ChIP-seq.
Exactly, using ChIP-seq data.
Mm-hmm.
And they found 45% of these new composite motifs were indeed more enriched, where you saw both TFs overlapping in the cell. It's real.
Wow. So this implies TF families are way more interconnected than we pictured, crossing family lines.
Absolutely. The analysis showed it clearly. TF-TF interactions commonly cross those traditional family boundaries. Some families, like TE-ATFs, were very promiscuous, partnered with lots of others.
Okay.
But even families we thought were less interactive, like C2H2 zinc fingers, they could form strong, important partnerships, like GLI and RFX3.
Which are important for?
Hedgehog signaling.
And forming cilia, those little hair-like things on cells, vital functions.
Okay, so tons of interactions crossing families. But what about specificity? Are these composite motifs just generic docking sites? Or are they picky?
That's what's fascinating. They're often highly specific. Really picky.
How specific? Close to half were restricted to, like, paralog groups or subfamilies, not just broad family interactions.
Remind us what paralogs are.
Right, so paralogs are genes that arose from a duplication event within the same species. So very closely related proteins.
Got it.
For example, the PITX subclass of HOX proteins, they specifically interacted with FOXA and FOXD TFs.
Mm-hmm.
And HOX2 paralogs, like HOXA2 and HOXB2, they specifically paired up with PROX1 and PROX2.
So even super closely related TFs can have totally different partners just based on the DNA sequence. That's incredible precision.
It really is, an extra layer of control. And what's maybe even more striking is how different these composite motifs look.
Different from the individual one.
Yeah. Analysis showed almost all composite motifs were clearly distinct from the individual TF motifs. Often, a single TF binds really weakly to that composite site if its partner isn't there.
Ah, so it needs the partner in that specific DNA shape.
Exactly. The middle part, where the two motifs kind of overlap, it often looks quite different from what either TF would prefer on its own. The ELF1 FOXK2 motif is a great picture of. This looks weird compared to the individuals, but the pair binds it strongly in cells.
Okay, this is clearly expanding the textbook view of gene regulation. But what about, real biology? How does this DNA matchmaking actually work in our bodies?
Right, the big question, does it have a real biological role? And the answer seems to be a resounding yes.
How did they show that?
They looked at pioneer factors.
Yeah.
Those are TFs that can kind of open up tightly packed chromatin kickstarting sulfate changes.
Okay.
And they found, well, some pioneers like FOXA1 are pretty promiscuous. They partner with many others, but other pioneers, highly selective. So it shows a really diverse interaction landscape for these crucial factors.
So these specific DNA-guided pairings could be key for how cells specialize during development, like how a neuron becomes a neuron.
Exactly that. They looked at the developing spinal cord and saw that pretty much all the specific cell types had their own unique composite motifs formed by the TFs specific to that cell lineage.
Like a signature.
Precisely. They found a highly paralog-specific interaction between HOX5 and OLA-G2, for instance. It really suggests this is how precise cell identities get established, how positional info gets integrated.
What about turning genes on, the activation part?
Good question. Many TFs with strong activation domains, the parts that actually recruit the machinery to transcribe the gene, like HOXB2, they specifically interacted with other TFs that maybe don't have strong activation domains themselves.
Ah, so a TF without its own on switch can still bring a powerful activator to the right spot.
Exactly. Through these DNA-guided partnerships and their in vivo experiments in mouse embryos back this up. Composite motifs like HOXA2, PROX2, or GLI3RFX3 drove really specific gene expression patterns.
Where you'd expect them.
Yeah, right where you'd expect based on known developmental roles, like in limb and neural tube formation.
Yeah.
It fits the biology.
And this isn't just some random quirk of mouse development, presumably.
No, definitely not. They found these interactions are evolutionarily conserved across mammals.
Meaning they're important.
Deeply important. If you look at data from the Zoonomia Consortium, that huge project comparing animal genomes, you see it. If one half the composite motif is conserved, the chance that the other half is also conserved goes way up.
So evolution actively selects for keeping these pairs together.
Wow. Okay, let's dig into the how a bit more, the atomic level details. This is where the method's really shown, right?
Absolutely. One of the key innovations here was what they call the cryo-EM-guided mutagenesis loop.
Okay, break that down.
They got these incredibly high-resolution structures using cryo-EM, especially for UTS-GATA bound to DNA down to 2.9 angstroms, almost atomic.
Incredible detail.
And that detail revealed something critical, a rigid kink in the DNA.
The DNA itself was bent.
Yes, in a specific way. And this kink physically forces the DNA binding parts of the TFs, their helix-turn-helix domains, to converge. It essentially nucleates the assembly, starts the process. So the DNA isn't passive at all. It's actively shaping the TFs, forcing them together, like a little molecular jig.
Precisely. Then they did mutagenesis, changing specific DNA bases, and combined that with SelectSeq to measure the energetic cost.
What did that show?
It proved definitively that it's the DNA backbone structure, that kink, that initiates the cooperative binding, not just protein contacts pulling them together later.
Mind blown.
And this insight was really helped by a clever technical invention by Jin Liu, a postdoc on the team, a DNA methyl kiss crosslinker.
What did that do?
It helped stabilize these complexes, because often these three-part assemblies, two TFs and DNA, can be pretty transient, hard to capture. This crosslinker helped hold them together long enough for the cryo-EM.
That's really clever. Okay, so what does this discovery that DNA structure is so key mean for computational biology, especially things like AlphaFold?
Yeah, that's a really important point. And it highlights a current, let's say, gap. Tools like AlphaFold and RosettaFold, amazing for predicting protein structures, even protein DNA to some extent. But they largely fail to predict the preferred spacings and orientations for these DNA-facilitated TF pairs.
Why is that?
Well, it suggests these tools, as powerful as they are, currently struggle with modeling these very subtle, very precise contacts, the ones that depend heavily on the exact DNA shape, orientation, and spacing. It's a different kind of problem.
So still more work to do on the modeling front.
Definitely. But this study gives them crucial data to work with.
So overall, this is positioned as, what, a third paradigm?
Exactly. DNA-mediated TF matchmaking. It seriously expands our regulatory dictionary, and it provides mechanistic insights that will absolutely inform future chromatin models, maybe even help improve those AI prediction tools.
And it's not just academic, right? You mentioned practical implications.
Huge practical implications. Think about the scale. These grammar motifs, these composite sites, occur maybe 50,000 times in the human genome.
Wow.
So this work gives us a clear roadmap for developing new therapeutic strategies. Imagine designing small molecules that could maybe wedge that DNA kink or smooth it out.
To fine-tune gene expression.
Precisely. To modulate that TF cooperativity. It could lead to entirely new classes of drugs.
Or gene therapy.
Your gene therapies, yeah. It fundamentally resets how we even think about controlling genes. And it gives synthetic biology an immediately useful toolkit.
Engineering gene circuits with more precision?
Unprecedented precision. They actually tested this. They did in-cell synthetic enhancer experiments, used CRISPR to tile libraries, introducing or removing these specific DNA scaffold sequences.
And what happened?
They saw massive changes in gene expression output.
Huh?
Over 100-fold swings, both up and down, just by tweaking that DNA sequence. It proves these sites have real power in vivo.
That's stunning confirmation, and the collaboration behind this sounds pretty innovative, too.
Oh, absolutely. It's a perfect example of interdisciplinary power. You had structural biology experts at MRC-LNB in the UK,
Right.
working hand in glove with functional genomics experts at the Broad Institute in the US, plus access to cutting-edge facilities like the EBIC Cryo-EM Center and massive sequencing capacity.
And handling all that data.
Cloud pipelines were essential, using platforms like DNXS. It allowed these iterative cycles, do an experiment, get the structure, analyze the function, refine the hypothesis, repeat. That just wasn't possible 5 years ago.
Amazing synergy. And you mentioned a machine learning tool earlier, the grammar extractor.
Yes, another crucial piece.
Yeah.
Developed by computational biologist Maya Scott. It's a transformer model, a type of AI.
What does it do?
It was trained using something called SHP-weighted commerce, basically, learning the important short DNA sequence patterns. And it can predict over 70% of TF co-occupancy events genome-wide, where two TFs are likely binding together.
How good is that?
It's about 30% better than previous models that just looked at individual TF binding motifs, a big leap. And they made the tool openly available so the whole research community can use it.
Fantastic. So stepping back, what are the big advantages for a cell to use these composite motifs, this DNA matchmaking, instead of just relying on individual binding sites?
Good question. The paper highlights 4 key advantages. First, combining two TFs lets the cell integrate positional information better during development, more precise addressing.
Makes sense.
Second, because individual TFs might bind weakly to these composite sites alone, it allows for dual functionality. A TF can be a partner here, but act independently elsewhere on its own preferred sites.
Flexibility.
Third, these high-affinity composite motifs mean the site can be occupied even when TF concentrations are relatively low. That increases specificity, Genes only turn on when both partners are present at the right place.
More control.
And fourth, maybe the most critical, it greatly increases the cooperativity of binding. Genes can respond much more sharply, like a switch, to small changes in TF levels.
Which leads to
More exact fell-fate decisions, sharper developmental boundaries. Building a complex organism requires that kind of precision.
truly groundbreaking stuff. So what's next? What are the big remaining questions or frontiers?
Well, a couple of big ones. Mapping how the local chromatin environment, how DNA is packaged in different cell types, affects this DNA flexibility in these interactions. That context matters. Right. And integrating all this with the 3D structure of the genome. How DNA looping brings distant elements together. That's still a huge area.
Putting it all together in 3D space.
Exactly. But this study gives us an incredible data set, a huge push towards cracking what some call the second genetic code,
Not just what proteins are made.
but the code that dictates when and where genes get turned on and off, the regulatory code.
So to sum up this deep dive, DNA is not just sitting there, it's an active choreographer.
Absolutely. Conducting this intricate dance of transcription factors, shaping the specific interactions that define cells, tissues, development.
It's a major shift in perspective, moving beyond just finding binding sites.
To understanding how DNA's own structure is the key, forming these complex regulatory partnerships. So you've now got the core insights into this new paradigm, all the fascinating details.
Okay, so here's something really compelling for you, the listener, to think about. If DNA really is this active matchmaker, this choreographer, how might understanding that dance let us do more than just fix genetic mistakes? Could we actually design new biological functions, reprogram cell fates with control we've never had before? What new possibilities open up when we see DNA not just as the blueprint, but as the dynamic conductor of life itself?